Hey everyone, here is a video on how to create a dihybrid Punnett square. So more notes on Punnett squares specifically for dihybrid inheritance. So that is the inheritance of two traits that are controlled by two different genes on two different chromosomes. So a dihybrid cross can be performed, and rather than a 2x2 box with a total of 4 squares, there'll be a 4x4 box with a total of 16 squares or possible genetic combinations or outcomes. All right, so let's get started. Let's start with the parents first, as we did for setting up monohybrid inheritance crosses. So in this case, we're going to stick to plants, just as Gregor Mendel, father of genetics, did as he crossed the pea plants and made observations about inheritance and patterns of inheritance. So in this case, a plant heterozygous for round yellow seeds is used to fertilize a plant that is also heterozygous for round yellow seeds. What is the probability that their offspring will have a wrinkled green seed or wrinkled green seeds? Note that round seeds R and yellow seeds Y are dominant. So we have a couple of different traits. So let's start with the different traits. So we have traits for parent 1. We have traits for these dihybrid crosses. And so because they're dihybrid, there's 2 traits. What are the 2 traits? Well, one is seed shape. And the other is seed color. So we have 2 traits. All right, so let's take a look at the phenotypes of the parents. So parent 1 is round and yellow. So round seed shape and yellow seed color. Parent 2 is round and yellow as well. So round seed shape and yellow seed color. Okay, so these are the physical traits or the phenotypes for parent 1 and parent 2. Let's use these phenotypes to determine what the genotypes would be for the parents. All right, so let's write the genotypes of the parents. So if we have parent 1, genotype, we have R heterozygous for round, and we have Y heterozygous for yellow. We also have parent 2. We're going to cross this parent, and we're going to fertilize the other plant. And this plant has physical traits round. Parent 2 is also round, heterozygous for round, and heterozygous for yellow. All right, so we have 2 parents being crossed with one another, and we have to figure out what are the genetic possibilities, what are all of the outcomes. So let's perform this cross. So we have a method that we use in order to figure out what the gametes are going to be, and this is called FOIL. So you may have heard FOIL. What it stands for is first, outer, inner, and last. And so this FOIL is kind of what we're going to be using when we figure out all of the different combinations of the gametes. Now, remember that gametes are reproductive cells and reproductive sex cells, and so they contain the alleles, and they include sperm and egg. So let's draw them out. Since there's 2 traits, and each trait has 2 alleles, okay, we're going to have a total of 4 gametes per parent. So parent 1, 2, 3, 4, crossed with parent 2, 1, 2, 3, 4. Now we're going to give these sperm some mobility, and those will be parent 1. All right, so we have to use our FOIL here, F-O-I-L, in order to create all of the different combinations for each of the gametes. So we have uppercase R, uppercase Y. So each of these alleles represents a different trait. So you'll have 2 alleles because there's 2 traits for each of the gametes. So we've got R, we've got uppercase Y. So that's first. Then we've got outer, uppercase R, lowercase y. Then we have inner, lowercase r, uppercase y. And then we have last. So we have lowercase r, lowercase y. And because this is also, parent 2 has the same genotypes as parent 1, then these gametes will have the same possible combination. So I'm just going to rewrite, but again we're using FOIL, first, outer, inner, last. R, Y, R, Y. Excellent. All right, so now let's go ahead and so notice that we remind ourselves of the law of segregation, which states that each gamete contains only 1 allele for each trait, so 1 allele for each trait, and there are 2 traits, so 2 total. All right, now they're going to independently assort themselves on the Punnett square, so we can figure out what are all of the possible combinations. Because the law of independent assortment says that the separation of alleles for four different traits is an independent event, so all combinations are equally as likely. So let's figure out what all of the combinations could possibly be. All right, so there are 1, 2, 3, 4, so it's going to be a 4 by 4 grid. So let's do that here. So we've got, here's a Punnett square. We'll do 1, 2, 3, 1, 2, 3. So that'll give us 4 by 4. So parent 1, again, will write their alleles across the top. And for parent 2, we'll write them along the left of the Punnett. Here we go. R, Y. Uppercase R, uppercase Y. Oops, that's a fancy Y. And parent 2. Okay, so now we have our Punnett square set up and we have all of our alleles in place, and now we have to figure out what are all of the possible combinations. So let's go. So we're going to cross big R with big R. What are some things to keep in mind as we want to write all of the alleles for the same traits together? So the same trait, R and R, for example. That codes for seed shape. And then we'll write the Y's together, and that will represent the seed color for each of the possible combinations. And then just a reminder to write the dominant allele first for a trait. It's regardless of which parent 1 or 2 expresses or has the dominant allele in its genotype. So just write the dominant allele first, just as you would write the first letter of your last name as an uppercase. Okay, here we go. So we've got 2 uppercase Y's. We've got R crossed with R, uppercase Y crossed with lowercase y, R, uppercase R, lowercase r, 2 uppercase Y's, and we'll continue. Those are 2 uppercase. Uppercase, lowercase from here. Whoops. The little eraser. Not very straight, but it works. All right, so we're crossing little r with little r. upper y, lowercase y, little r with little r, 2 little y's. We've got 2 little, we've got an upper r, uppercase r with a lowercase r, and we've got lowercase r, uppercase, lowercase r, lowercase y, lowercase r, uppercase lowercase y, lowercase r, and 2 lowercase y's. All right, so that's our all of our possible genotypes for this specific cross, and so we're going to write down all of the genotypes. So let's do that. So we've got uppercase R, uppercase Y, uppercase Y. So we have homozygous dominant for both seed shape and seed color. I'm going to write down all of the possible combinations for the genotypes. And I'm just going to write one more over here, little r, little r, little r, little r. So we have 9 total combinations. All right, so we have 9 total combinations, and we're going to just have to do one more step, which is to figure out, the probabilities, phenotypic ratios, and percentage of offspring that have each of the traits. So let's do that on a different page. Get it set up, and we'll flip back in just a second. All right, so we have, let's see here. We have phenotypic ratio, so here are the results. And we want to get phenotypic ratio. All right, so this is for 2 traits. So we want to know how many round and yellow. I'm going to write a little smaller. Let's see here. So we need to know, whoops, round and yellow. We want to know wrinkled and yellow, and it's a ratio, wrinkled and yellow. We want to know how many round and green. And we want to know how many wrinkled and green, which is what's left. Excellent. All right, so here's the phenotypic ratio. We're going to have to go back to the second page, and we're going to have to count. So, for example, for the first R, big Y, big Y, that would go on to, so this genotype would predict that the phenotype would be round and yellow. So any of these combinations that include an uppercase R and an uppercase Y are going to go on to produce round and yellow offspring. So let's count them up. So we've got, if we can highlight here, is blue. We've got 1, 2, 3, 4, 5, 6, 7, and 8. We have one more here, 9. So we have 9 that will result, are predicted to result in 9 out of 16. So we'll write that there, 9. Let's go back and count how many wrinkled yellows. So we would have, the wrinkled yellow would have 2 lowercase r's and at least one uppercase y. So either of these genotypes would, so let's go and pick a new color. Let's pick this color. All right. And so we're looking for either of these 2 genotypes here, for wrinkled and yellow. Let's do it. Wrinkled yellow, wrinkled yellow. And I think I found a mistake here because I noticed that this is an uppercase y and I wrote a lowercase y here. So I'm just going to correct it. That should be an uppercase y. And that would be our third combination. So we'll count 1, 2, 3. Let's go back and update that. Whoops. Let's advance forward and update that, I should say. 3. How many round and green? Well, let's check it out. So if we look at our phenotypes for round uppercase R and 2 lowercase y, uppercase R and 2 lowercase y, it looks like there's 2 different possible combinations here as our options, and let's find them. So we've got 1 uppercase R, lowercase r, lowercase y, lowercase y. So we've got round and green, and we also here would have a round and green, and here would result in a round and green for a total of 3 possibilities. And then last but certainly not least, we have the wrinkled green, and we'll just pick a pink or a fuchsia for that one, and let's go back and count. 1. There's 1 left, so we'll update that. So we end up with a classic Mendelian phenotypic ratio for a dihybrid cross, 9:3 to 3:1. So let's go ahead and answer what percentage of offspring have both recessive traits. So if we go back and look, this is both recessive traits, okay, right here. So in order to calculate the percentage, 1 out of a total of 16, because this is always going to add up to 16, and 1 out of 16 times, we want to get it into a percentage, 100%, then we could calculate the percentage of offspring that are predicted to have both recessive traits, and that answer is 6.25%. All right, and that is an example of dihybrid inheritance and how you set up and complete the Punnett cross.